Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonise approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
For all the input that we received, and I hope that I can go through this. In case you're wondering, I am intentionally not in presenters mode, so I can jump because some people will text or put in the chat that they have to leave early, and we have quite a few presenters today. So without further ado, let's get started. There's a couple of updates from the FDA, and Brandon, I don't know if you're ready now or if you want me to go through a few slides.
I'm ready now if you want.
Yeah, why don't you start then?
I don't know if you want me to share my screen. I'll put the links in the chat and just name the titles of the things I'm wanting to announce. There is a program here at CDRH, Center for Devices, called the Centers of Excellence in Regulatory Science and Innovation, called CERSI. And there's right now 4 centers out there. Some of our partner organizations, 2 or more groups entering together. But this program has been going for, I think, 4 or 5 years, and they are putting out a call for proposals soon. And it's an opportunity to get funding for research on regulatory science issues that has a strong partnership with folks like myself and my colleagues here at this center, and opportunities to share. And there's a agreement framework and stuff that encompasses that. I'll put that in the chat. The other thing that I wanted to tell you, there's a report on the software pre-certification program that some of you may have heard of, where the idea was to kind of qualify organizations according to their quality management systems to hopefully impact their status as a submitter of devices and algorithms, and hopefully make a pathway that's less burdensome. So there's a report on that. That pilot study is over, and the report is written. It wasn't wholly successful to go in that direction, and the reasons why and some of the background of that is included. And then the third thing that I wanted to announce is that my office, the Office of Science and Engineering Laboratories, has reconfigured their regulatory science tools catalog. which I think before was mainly just a list of papers and GitHub sites with software and things like that. Now there's a little bit more structure and rigor around what we are calling regulatory science tools. And my software related to the webinar I hosted with, or ICC hosted, is one of the 5 inaugural tools of this redo. And check it out. I think I feel pretty excited about how they put it on the FDA website in a pretty fabulous way. And I got a DOI number, so now I can point to all this stuff. Those are my things.
Wonderful.
I'll put them in the chat.
Yeah, I know I had the CERSI, the regulatory science tools. I have the links, but yeah. And you can still see my screen, correct?
Yes.
Perfect. So the next item that I will drop in the chat is a document, PDF, outlining, comes out every year, focus areas of regulatory science from the FDA. And for those of you who are interested, when reading through this, 2 elements came crashing down, which is very good, which are technical factors influencing AI reproducibility for digital pathology applications. Look at that. And methods for assessing the generalizability of AI performance in digital pathology applications. Now, if this is not good news for this group, I don't know what is. I mean, it's clearly outlined in an official document as a focus area for regulatory science. So I think, of course, if you're interested in, portraying this in your respective societies or groups, it's clear that these 2 elements, technical factors and methods, will definitely be, of high interest. Another update, which is today, like it's from today. So 9/28 is the final guidance document on clinical decision support software. I put the guidance in the chat, and as you will see, there's also a link to it if you want to use it, but it's a very frequently downloaded document. And I want to briefly outline one of the things that is, an interpretation according to a section 520.01e, etc. And that is what is considered not a regulated device, which when it comes to software and in terms of clinical decision support, there are basically 4 criteria that I outlined a bit here. This is unfortunately cropped, but basically there are 2 to 3 things. It is, if signals are processed from an IVD, that would be a criterion that it is considered, a medical device and would require regulation. If there's a user using it without any other input, that's not considered correct. And then the last part is that there has to be multiple sources. This is, of course, a large oversimplification. Please refer to the guidance document in the chat. There are many other components in terms of clinical decision making, but I just wanted to show that this is very clearly defined. And in contrast to the prior versions, this is now already dropped in the Federal Register. And I think it was actually finalized yesterday, but official date of release, which is also with a bit explanation, as you can see here, is 9/28. And, just make good use of it and consider, applying these rules. One element that 2 people emailed me, I'm not sure whether that is applicable to everyone. It was a disaster a few months ago, and there's a final evaluation document about the infant formula shortage and response. I think the part that was very interesting in the report, I just dropped it in the chat, is good manufacturing practices and supply chain elements that were addressed. So without further ado, I just wanted to mention it because 2 people emailed me about it. A quick overview over what can be best described as probably something like a legislative rodeo, or at least a bonanza, which is, summarizing a lot of things that are happening. If you haven't seen this, there's 2 organizations lobbying for certain cost-related aspects related to PAMA and the implementation. If you haven't seen that, I'm just pointing to it. One interesting House passed the bill. One interesting bill is to install electronic prior authorization for Medicare Advantage plans. Prior auth is a huge burden. I hope it will not come soon for anatomic pathology, molecular pathology, and comprehensive genomic testing. That's the case already. But the requirement, legal requirement, that electronic prior auth is relevant, please take a look. Now I point to a fantastic blog by Bruce Quinn, who took the time to compile all the relevant legislative changes that the 107th Congress is dealing with, and he called it a bonanza. You may have heard about Valid Act. Next slide will talk about this. SALSA Act, which put guardrails around PAMA. We just mentioned that. But there's a few other acts that I just wanted to throw out there that people have heard about it. One is that there's a Medicare Multi-Cancer Early Detection Screening Coverage Act of 2021. Basically, in quick summary, when it's FDA approved, Medicare should cover that. There's another Medicare Act, which is called Reducing Hereditary Cancer Act. Another Medicaid and Prenatal Genetic Screening Act, which is called Expanded Screening Access Act. The Precision Medicine Answers for Kids Today Act, which is a PEDS ICU Genomics Act. Right Drug Dose Pharmacogenomics Act. A Patent Eligibility Restoration Act that would exempt certain diagnostic test patents from not being available. And multiple additional acts at the state level that passed and incentivize biomarker legislation. This term here, I came across for the first time, but that's apparently a thing. If someone would have told me that 2 years ago, I wouldn't have not thought that there's biomarker legislation, but so be it. And then one interesting new institute that I want to outline, which is a Translational Medicine Institute under the NIH. Quickly, you may have heard already, several news outlets reported that the Congress passed the FDA user fee bill without the Valid Act. You see that is currently, I think, still in the making. There are still other ways that the Valid Act could be attached to the current legislation in sort of an omnibus address or approach, but that remains to be determined. But for now, that is removed, and the consequences of that are obviously manifold and remain to be determined, but it's probably important for this group to know because it is affecting many of the high complexity tests. The other component that I wanted to mention, which is a new US Department of Health and Human Services under that HHS setting, it's called ARPA, which is the Advanced Research Projects Agency for Health. Some of the goals of this particular branch of the government now is translational medicine research to specifically focus on speedy application of health breakthroughs, support user-driven ideas, and you see that there's also a DARPA-related element with defense, probably related to infectious diseases, etc. I wanted to point this out because some of the mission statements, the news and publications here, are happening, and I presume that there will be a hiring string starting soon because the funding came directly in relation to the NIH funding from the Biden administration as well as through NIH and the executive branch, Moonshot, etc. So definitely something to look out for, especially when it comes to integrated multimodal digital health solutions. Okay, the next section of today's meeting is payer strategies. There's a few interesting things out there that I wanted to mention. The first one, again, from Bruce Quinn, I share this quick link here with everyone, which is a so-called scorecard that is developed by Medicare for coverage of emerging technologies. It's a great summary, 5-minute video. If you want to learn more, please click the link, stop it now, and listen to it later. One very interesting paper I wanted to share is by a group that have maneuvered something that you rarely see in peer-reviewed publications, which is the actual dollar amount across hospital cash prices and commercial negotiated prices for 70 services. I dropped the paper in the chat. Please take a look. It is definitely relevant, especially because it's CMS specified high volume services, and seeing that is very, very interesting. many of you have heard, the payer strategy work group has been very effective, had several meetings in relation to the digital path codes, and it's a pleasure to have Laura here. Hey, Laura. Can I hand it over to you for a quick summary? I think the slide from the payer strategy work group should be up now.
Awesome, great, thank you. Yeah, so we were took a little bit of a break, but picked things up again this year and are planning on resuming our full schedule of activities, including getting I think bi-weekly, maybe monthly working group call on the calendars. So those are who are interested in either resuming or joining those working groups on payer strategies, please reach out to Joe or Ula. I'll volunteer them to make sure that you get put on the calendars. So where we left off previously was, we had developed a strategy on pursuing obtaining reimbursement for digital pathology. Part of that was a larger strategy on just sort of how to get those payment codes approved. But the other side was also thinking about, how can the industry work to generate evidence to support development of new codes and also just reimbursement more generally. So since reconvening, there are now CP codes that have been generated that are category 3. And so we recognize that category 3 codes are less appealing to use simply because they don't come with a higher reimbursement level. But also recognize that using those codes are extremely important to generating the data and the evidence to support, moving those up to a higher category and attaching a real dollar reimbursement to those. So we realized that we need to do some education. Basically, is the short answer for our strategy. So we're working to sorry, I just got distracted. So sort of a twofold approach is encouraging more use of those CPT codes to then be able to collect more data and generate more evidence. So we are working between the Digital Pathology Alliance and CAP to record a podcast taking place at Pathology Visions to educate on the topic. We will have a PICC webinar to create awareness and understanding around this. What's the current landscape, using some examples of what's going on and how to develop more codes. And really that's going to be generated at trying to create some momentum towards using and getting everyone actively involved. And then last, pursuing some potential collaborations with the global DPA task force to more globally align efforts on reimbursement strategies. So that's generally what we've got going on right now.
Thank you. It's quite a bit. I think the podcast as well as the webinar will also deal with the practical aspects of implementing this category 3 code, and thank you a lot. Esther and Laura have pushed this forward. I see Eric is on the line. There have been several people, Charles, I think probably on the order of 6 or 7 that collect all the data, and we hope that the webinar will happen likely after Visions, so probably at the end of October, and hopefully we can help people roll out this category 3 code as soon as possible. Would be great to coordinate this with CAP as well. I see Mary and Monica on here. I think there's probably some initiatives through CIP as well, but any help, any way, any shape. 2 other quick resources related to the payer initiative. One thing that has happened for a lot of molecular testing is this ESMO-based scale for clinical actionability. I know a lot of the biomarkers, a lot of the applications of digital pathology, especially when it comes to quantification, have reached, I would say, a layer that goes beyond a hypothetical target. Several quantitative biomarkers, for example, Kia67, have been FDA approved, so we're definitely moving way beyond the investigational level, but more into the level that it's almost ready for routine use. So I wanted to point this out as an article. There's a very brief summary that I put in the chat. If you just Google this, you will find it's a whole framework that I believe is very helpful. You see it's published in 2018, so ESMO follows this quite succinctly, and I thought it may be a great way to think about, some of our applications. Another paper that was definitely of interest, and I wanted to share it, which is a discussion that we had several times, which is the cost versus value debate. So, if digital path is very valuable, you can just implement it. Question is, why do we need separate reimbursement, and how can we follow this? And this brief paper outlines this exact debate very well, including any quality related aspects that may matter in that context. All right, quick switch from the payer group to MDIC. For those of you who haven't met Noor Falah, I think Noor, you're on the call, correct? I think we have an MDIC update, and I hand it over to Noor.
Hi everyone, my name is Noor. I recently started at MDIC about a month and a half ago. And just some quick updates about MDIC. We've been working in collaboration with the FDA, NIST, NIH, and a few other organizations on improving the accuracy of next generation sequencing. So that was a big product that we were very proud to be in collaboration with a few other organizations. Next slide, please. And then on September 12th, we had the Cybersecurity Summit, which was our first in-person event after COVID lockdown. So it was a great networking opportunity, and we had a lot of great positive feedback. Recordings will be available in a few weeks. We're just waiting on some legal stuff and all that mumbo jumbo, essentially. Next slide, please. And then our next initiative is the threat modeling boot camps. If you are interested in joining, please either reach out to me. My email is, I'll type it into the chat, or you can right there. Thank you. My email or Jitesh. And the cybersecurity initiative is very important to us, and we are very excited to be launching the threat modeling boot camps. The previous year, 2021, I was obviously not a part of it, but it was in collaboration with the FDA. This time around, it will be in collaboration with Shostak and Associates. And as Joe is pointing on the screen, there are two of them. They are both virtual. One will be held towards the end of October, and then the second one will be around the middle of December. Both are going to be very intense and will require a lot of seated time to do homework and participate in classes. So if you're interested, please join us. We would be more than happy to have everyone join us. And I believe that's it.
Yes.
All right.
Thank you, Noor. Great updates. I don't have the paper right now. Noor, if you have the leveraging paper, if you can paste in the chat, that would be great. I just can't find it at the moment on my desk.
Sure.
The next element related to our circulating tumor DNA initiatives, which is related to a draft guidance document that was reviewed. We have this as a project currently housed under PICC. There are many different projects going on, of course. I'll paste the chat here real quick, which is like a summary of the draft guidance and additional information. In relation to that, we had several meetings. You find everything on the link that I just pasted, and there was a paper that is spearheaded, or at least in large parts organized by Friends of Cancer Research. I pasted in the chat as well. So one of the things that seems to crystallize out of these numerous different value streams related to ctDNA is that circulating tumor DNA seems to be a different endpoint. So instead of having, objective response rates by radiology, it could be that the clinical benefit is actually measured as an outcome using ctDNA. That's sort of a slightly different approach to demonstrate the value of a new technology, but this is definitely related to immune checkpoint inhibitors, a great paper. So please take a look, and high five to our friends over at Friends of Cancer Research. Two additional resources provided in, I would call it, the large topic of diversity and inclusion. One is a fantastic resource put together on the so-called Nordic model of education. So for all the educators among you, I think many are involved in resident or fellow teaching, but also other ways of teaching. I know Brandon has a lot of students and rotating people that he educates frequently, and this is a book. It's open access and outlines at a scientific level how to account and how to optimize diversity in terms of education. I thought it was great. I'm not through. It's like a couple of very intense scientific papers, so I just thought I'd share this. The next element is a cross-sectional study. Rare. The design is always controversial, but I thought this is in the Annals of Internal Medicine. And speaking of diversity and inclusion, you may have heard about the issue related to racial bias in glomerular filtration rate estimation. And I haven't seen a US community-based epidemiology cross-sectional study on the topic. This is a very well done study, and I thought that it serves the purpose, at least in terms of informing from a scientific perspective. It doesn't replace a meta-review of prospective trials, but it's definitely a great study. A similarly fantastic summary actually comes from the Office of Inspector General, and I pasted the publication here as well, which illustrates a interesting pattern of utilization of telehealth during the first year of the COVID-19 pandemic, which utilizes HHS and CMS data to demonstrate, as you can see here, that urban and Hispanic beneficiaries apparently had a higher likelihood of utilizing these services. It is an illustration of telehealth, so remote diagnostics falls into this. Unfortunately, data is not parsed out in that aspect, but it's definitely, if you're interested in remote work or telehealth, this is an important resource that just came out a few days ago. Switching to a couple of resources. Definitely, again, a very active month since last month, and I wanted to give a big shout out to the Longevity Group. It's a big patient and oncology active group on numerous layers. One element that they brought out, which I think is a fantastic way to educate patients, it is how to read a biomarker test report. I know many people in pathology laboratories have conceptualized, at least after the information blocking went live, to consider brief summaries on lay terms, 8th grade readability indices, how do you actually communicate something to a patient who's not an MD, who doesn't have the background. And this is a way to outline for lung cancer how to read a biomarker test report. Definitely a very, very impressive resource. Another resource I wanted to share is, again, Friends of Cancer Research. You see this group is very active and pulse of time. The link is in the chat, and I want to show you this quickly. And this is a great overlap with our friends at the FDA. So this is a searchable or downloadable interface that lists all the sponsors, the years, and all areas of, for example, cancer or rare inherited disorders regarding breakthrough designations. It highlights those that you pre-select, and you get basically the specific indication. So it works very well. And if you have an internal database that you want to subset, that is, fairly active, works very seamlessly, and it's frequently updated. So this is a breakthrough therapy database maintained by Friends of Cancer Research. So for molecular pathologists or other people interested in breakthrough therapies, this is a great way to organize this data fairly quickly. Not sure if Brittany, who's representing Friends, is here and want to say anything. Maybe a little bit later. One group that we're collaborating with now with PyCC is ARHQ, if you're not aware. That's the Agency for Healthcare Research and Quality, another branch agency under HHS. And they are very interested in identifying diagnostic or laboratory safety errors. As part of that, they provided a resource, which is a 57-page PDF. They call this a MeasureDX resource, and I put the document in the chat. What they are proposing here is a so-called safer diagnostic framework. You see sort of a pictorial representation here. It's actually, the concept is not new, but it has been published several times, but it has now been adopted as part of the proposed avoiding diagnostic errors in quality management systems. So the resource itself is a few pages. It's an easy read. It's a great resource in case you're looking for quality improvement initiatives, etc. In the same context, also another resource that we collaborated with this group. They offer CHCQM board certification, which is a healthcare quality management certificate. I have reviewed the individual criteria that they apply to get this certificate out, which is covering a lot of the aspects under CLIA, a lot of the components of a quality management system. They offer educational seminars and, again, this sort of healthcare-focused quality management system. They are not ISO compliant in all aspects, but I think it's definitely a resource to look out for. And, it's definitely, helpful to have that. And I hope the link hopefully works. A few more resources. One came from a group in Brazil who published Nature Medicine, I believe, 10 high-yield recommendations. The focus is, of course, in Brazil, but it's applicable beyond that. This came from the Oncology Task Force, and they provide very concrete elements, including diagnostic focus when it comes to oncology and realizing cancer care. So that's a very interesting, very short resource. I think 3 pages, 1 table, so please take a look in the chat. Another group around Rodrigo Dienstmann is proposing this approach for a so-called wayfinder, for those at NASA and Space Fan, definitely a breakthrough. But here it is a global precision oncology registry. And again, the theme here, similar to the last one, is to integrate data across a large series of platforms and or territories. And the resource is available. The framework is portrayed here briefly, but there's a little bit more to it, so I thought I definitely wanted to feature this. Staying with the data models, we have one additional paper here. This is currently in preprint, but I think this will make definite impact here. We have a substantial problem in organizing, and the fancy word for that is ontologizing healthcare data in a systematic fashion. You could call this whole thing just a data model. But as you see, what they tried to depict here is the data in typically a non-relational fashion has to be put together. And a lot of people have portrayed various ontologies. The problem is demonstrating immediately that you can do translational discovery usually is missing. And this paper around Tiffany Callahan and Michael Kahn's group does that. or does that, has demonstrated it. And I don't know where this paper will show up eventually, but look out for it because it's very well done, and you know the discoveries sort of in healthcare data jungle are quite impressive. Few more resources. Of course, they're all publications, but I think it's quite impressive. One review here about teaching medical device design using design controls. I think this is covering a lot of the topics like calibration, reference materials, design control, etc. So definitely helpful. And a review about immune checkpoint inhibitors that I thought was very informative. So group about Emily Lin, etc. And I think this came yesterday from Lorenzo Bonaguro about just system level immunomics. If you look at the figure, it probably gives an overview over all the various components that you need to consider when working in systems immunology, which is apparently a booming field, as you probably know through all the immuno-oncology threads. Since we switched to papers, it is super amazing. I will try to share all the papers, but I drop them all later, and you can download those, or they will show up on the website eventually. But I wanted to show and highlight I think 10 or so that are really impressive. One was spearheaded by Dr. Baez Navarro and Roberto Salgado, which is how to select HER2 low breast cancer patients. I'm sure you saw the approval. It is quite a tricky subject, and this is a SWOT analysis, so strength, weakness, opportunity, threat. Short paper, easy read, but definitely important. This is a paper that occurred in Nature, specifically the subsection News and Views Pharmacology. This sort of two novel compounds can avoid a drug toxicity, which gets into a very convoluted regulatory environment when you need two drugs to make something safe. We got several questions about it, whether that's applicable. I thought I'd share it because one of the elements is, of course, the preclinical assessment for toxicology relies heavily on microscopic examination of animal tissues, and we have a CPIM project with several groups from Europe and the US working on this, so quick shout out to this group. There's a paper on somatic variants in normal tissues. I leave it at that. Extremely controversial. This is in the Annals of Oncology, just came out a few days ago. Extremely impressive. And with that part, which is sort of genotyping level, we switched to AI. And there's quite a few things that reached us. One is an image-based fusion detection from slides poster that was presented at ASCO. You can, if you want to take a screenshot, or again later provide this. Quite impressive sensitivity and accuracy over quite a few cases, and this is a rare entity, so definitely impressive. There are two AI comments, one from Jonathan Chen and Dr. Dhaliwal, and the other one from Dr. Reina and Snousey, if I pronounced that correctly, discussing diagnostic performance algorithms and the decoding for diagnostic excellence. Share both of these quickly. I wanted to do two somewhat slightly deeper dives into these papers because they're like absolutely phenomenal. JAMA Dermatology, this is a real-time analysis of using 2-photon fluorescent microscopy, and the setup is as follows. So this is the portable system, and the intent is that an excision is stained for 2 minutes, mounted, and real-time evaluated, while subsequently processed for FFPE. So far, probably nothing fancy and or new. However, when looking at the portrayed images in the paper, I was astounded. So the top part is the original, so that's the bright field image. The bottom part is the 2-photon image that is basically immediately obtained. Now I would consider that diagnostic rate. That's just my opinion. I think, you know, having this published is definitely something very, very impressive. So this shows that other imaging modalities, again, in this case, a two-photon fluorescent microscopy can result in almost, or if not substantively similar images to real world histology. So I thought I shared this, so definitely great
Was that
work.
on dissected tissue or just
This
intact
is
tissue?
intact tissue that is mounted and evaluated. So it's basically a virtual section using a two-photon microscope through an intact piece of tissue. It's scanned. It's cool, right?
Look, it looks better than OCT.
Well, I thought, you know, I mean, there's definitely some differences, for example, in this case here, in the crowding and, you know,
How did they get the stain to penetrate the tissue?
I mean, it's, you know, it's skin excisions, so those are just, you know, a few millimeters, so I presume that that works, you know. It may not work with every single specimen. I don't know if they did sort of a serial assessment, but according to what they present in the paper, it works. So, you know, as they show here, I don't know if this is an intradermal excision or shave biopsy, but, you know, if it's just a few millimeters, apparently 2 minutes is sufficient. But I don't know, that would be probably a question for the authors, but great question. A similarly interesting paper came from JAMA Surgery, where a device is used to identify, this is a small probe, and you know, this is using reflectance spectroscopy, so another different imaging modality, or one should say sound modality in a way, where they use this pattern of spectroscopic signal to distinguish tumor versus non-tumor tissue. I will share this. It's, I think, a group in London, and definitely also like a very interesting approach. There's several papers about multimodal biomedical AI. This is a review from Eric Topol. I think the next step is, of course, that, you know, multiple imaging modalities come together and how to discuss that, and, you know, definitely the term multimodal AI crossed at least my desk in reading all these resources quite a bit. Without going into too many details, there were 4 or 5 other additional papers that I will share with everyone, but one that I wanted to definitely mention here, especially with Brandon on the line. This was published in Modern Pathology, Meyer et al., a direct identification of ALK and ROS from NSCLC, H&E slides. I mean, definitely a great paper, great figures. I think it's a proprietary algorithm to say it. You can read it, but the part that I thought I wanted to point out is that the summary of the data illustrates that they confirmed this with 6 positives and with 2 positive cases, and determined that the sensitivity in identifying this is 100%. So I would consider this a proof of principle, so it's definitely on the research side. I'm not sure whether this would suffice the predicate of clinical validation, but I wanted to point out that, you know, this seems to be at least sufficient for publication. At least it shows that there's a lot of interest in the field here. Another AI tool that I thought is very interesting, and I wanted to show this, I hope that this will work, is called the Biotech, from the Institute of Research in Barcelona. And the interesting thing here is that you can actually ask real-world written questions and then focus on individual components of a question. So what drugs target diseases with specific genes? You can erect these sort of conceptual frameworks to then download specific data and components. This is just the tutorial, meaning the online version that is available. The source code and the individual subsequent databases and components that are in there are linked. So every single, you know, drug bank, etc., is linked here. So you can, you know, jump to the resource that provided this particular component. So I think why I wanted to mention this, it was accepted at the end of August, just after our last steering committee meeting, is that we're apparently now reaching the stage where you can ask at least conceptual questions and draw from a whole array of biomedical data sources. One paper I wanted to mention more in the radiology space, but I thought that 2 or 3 elements that were interesting here, same context, is the vision and text transformer to ultimately use both as so-called similar elements to train the one or the other, either to train from the text what the image looks like or from the image what the text will look like, i.e. for generation of this. This was done from unannotated chest X-ray images, and it's a great, study. Great paper on spatial transcriptomics. Fantastic paper on melanoma, uncoupling growth versus metastasis and identifying the cellular subset or niche that goes into the bloodstream. And I hope that this wasn't total overload, but I will share all the papers in the chat in a minute. Quick follow-up on a couple of events. The first one is from the National Academies of Sciences, Engineering and Medicine. They are starting a roundtable on genomics and precision health. I think the specific date for the webinar is October 12th. So that's definitely, around visions, but definitely before our next meeting. So if you Google Roundtable on Genomics and Precision Health or National Academy of Sciences, you will find this. It's for free, and I believe it's 10 a.m. I think it's planned for an hour or two with a lot of interesting discussion points. Oh, sorry, I prepared this so I can share this real quick. And then, of course, Visions meeting, I mentioned it before, so DPA organizes this October 16th to 18th. And so with that, we reached the official end of this steering committee meeting, but as always, I open it up for discussion. And please.
Can I say something on the last slide you showed on Pathology Visions? I'm going to be there and would enjoy connecting with anyone in this group while I'm there. So if you're going to be there and you want to connect, go ahead and either find me or shoot me an email and we can set up a time and have lunch or something like that.
Oh, absolutely. So I see there's a few questions in the chat. The I/O review. Yep, I will share all of the papers momentarily. Just a little bit tricky right now to share each one, but I will definitely get to it. Give me 1 second. Other questions, suggestions? Any news from CAP?
Thank you. I appreciated the update of discussing the need to have people using the digital pathology CPT codes. I agree. We need to do those for DigiPath and AI. And that initially, while it will have a lot less impact on our bottom lines, it will definitely have an impact at CMS for there to be an understanding of the breadth of utilization and the chances of it having more economic impact down the road. So CAP is working simultaneously on some initiatives to make sure that our membership understands that this is also a growing, important, and actual, like a now situation that so that we can get more people using these codes and strengthen everyone's position.
Oh,
And I,
absolutely.
yeah, I just wanted to say, I heard this morning that there was going to be an announcement today between SNOMED and Loink, and a new agreement has been reached. It was supposed to drop at the SNOMED annual or at the expo today. So I don't know if anybody's heard or if anybody is there, but look forward to that. A lot of that stems, I think, from the SHIELD work, or will have something to do about that.
Would definitely be great, but I haven't.
So it's on their website.
Oh, it is now? Okay.
Oh, no, wait, that's the epilepsy. Sorry.
Yeah, yeah. I had just checked before this meeting and I didn't see it yet, but yeah, keep looking.
Perfect. So I think the two things that are coming up is, we're currently looking for potential speakers. I know we had a prep meeting yesterday with Eric and the payers group about really the practical implementation. I see Joe Sirentrappen is on, so we're looking to put together a mini webinar probably towards the end of October, when I think the first institution have realized a laboratory information management based or HL7 based or FHIR based way to integrate category 3 codes. I know in our healthcare system we have our coders ready, but getting basically gap stops in place that the procedure has to be documented so that the coders know that scanning was performed. I think currently it's 12 or 13 codes that cover different levels, i.e. different effort levels for I think 88304, 5, 7, 9, etc. plus enzyme histochemistry, immunohistochemistry, and quantification, including ISH. And then the question is, of course, where should this be documented, how it should be documented, and whether you need to mention the scanner, whether you need to mention how many scans were done, like all of these practical things we want to cover in sort of a brief webinar. So if anyone is interested in speaking or sharing their own experience, please reach out to me or Ulla, and then we can solicit that and put together a mini agenda for a meeting. Okay. If there are no more questions or comments, I wish you a happy Wednesday and hope to see you next month at the end of October. Bye guys. Thank you.